Hello students, welcome to Aakash. I'm your botany faculty, Dr. Sukirti, and today we are here to discuss this particular paper, mock test 9 of AIATS PLUS 2025, phase 2, which was held on 16th of March 2025. So in this particular session, we are not only going to deal that how you're going to solve the questions, but also going to talk about the entire periphery of the topic so that you can know the fine nuances of those topics as well, right? So in this particular session, there are certain chapters that we are going to cover, and those are going to be principles of inheritance and variation, cell cycle, microbes, organism, and not organism, but ecosystem and respiration in plants. So good morning, everyone. So let's start with this particular session, and the very first question, that's question number 103, and it says that mitosis is called equational division because, right? So when we talk about this particular division, mitosis, right? So when we talk about mitosis, we know for a fact that if we are taking, let's suppose, this is the, mother cell, then the daughter cells that we are going to achieve after this will also be going to have the similar sort of chromosome, right? The chromosome number will remain the same. So we know that if the ploidy is 2n in the mother cell, the daughter cells will also have the same ploidy, and because of it, we say that the mitosis is the equational division. On the contrary, if we talk about meiosis, then whatever the ploidy of the mother cell is, exactly half of that ploidy will be received in the daughter cells, right? So Sagnik is correct, who's saying that daughter cells have same number of chromosome that of parent cells. So yes, this is absolutely true, and this is because of the fact we call mitosis as the equational division, right? So getting back with this on the question, so the first one is saying that the parent cell is divided into two daughter cells, right? So this is a main objective of the cell division itself. Whether it is mitosis or meiosis, either way, the cell is going to divide it into the daughter cells. Like after mitosis, we get two daughter cells. Similar way, after the completion of meiosis 1 also, we are going to receive two daughter cells, right? So this is an incorrect option. Then second one is saying daughter cells have the same number of chromosome as that present in parent cell, as you have already, discussed that daughter cells going to have the same ploidy. Because of that, we are going to call it as equational division. Third one is saying that two daughter cells so formed are identical in shape and size. So this is again incorrect. This could be the outcome, but because of this, we don't call it as equational division, right? Because of this particular feature, mitosis is having a significance of cell repair, right? So similar sort of cell could be achieved again if we are going to have this particular division. Then we say all the cell organelles are equally distributed to two daughter cells. Again, this is incorrect. Why? Because this is the main objective of the cell division, because when during the cell division, firstly cell growth occurs in order to prepare itself for the cell division. So whenever this growth occurs, because of the synthesis of many molecules in the cell, that's when the nucleocytoplasmic ratio gets disturbed, right? Or जैसे ही ये ratio disturb होता है, the small nuclei is not capable to control the activity of the entire cytoplasm, and that's how the cell gets the signal that this is the time to divide, right? So after that, it's paramount that whatever cell content is being present should be equally distributed into the two daughter cells for the proper functioning of the organism. If any of the mishap occurs, in that particular case, that cell will be showing certain sort of anomaly, right? It could go to the cancerous path, or it could be killed because of certain checkpoints pathway. So this is again incorrect. So mitosis is called equational division, and it is correctly depicted in option 2 because of the same ploidy level, right? All right. So moving ahead, we have another question, and that's question number 104, and it says that the final stage of meiotic prophase 1. So when we talk about meiotic prophase 1, right? So talking about the prophase, we will be having leptotene, it begins with it, a small lived stage, right, over here. So why fourth is wrong? In the previous question, that one is wrong is because the equal distribution is again going to be observed in meiosis as well, right? Whatever organelles you're going to have will be equally distributed again. That's the reason. And also that is the main purpose of cell division itself. It's not that in mitosis only, this is happening. Okay? All right. So getting back to this one, prophase 1, leptotene, it begins with it, very small lived phase. So in this particular one, the condensation of chromosome, a very paramount feature, a very characteristic feature of prophase is the condensation of chromosome. So that's wonderful. So in this particular phase, that condensation begins, right? Then this is being followed by the stage known as zygotine, right? Name itself suggests zygote. Usually when we call this particular one name, the zygote, because of the fusion of two things. Over here we are not going to fuse, but we are going to pair up the two homologous chromosomes that we have with the help of synaptonemal complex, and this pairing will be known as synapsis. So the key event of zygotine is synapsis, and here is the beginning of condensation of chromatin material. Then further, after these two stages, the main event is going to take place because of which meiosis is so useful, and that is crossing over. So pecitine is the stage where we are going to see crossing over, right? And this is going to be accomplished by the recombination nodule, which is having the enzyme recombinase, right? So there what we are going to have, we will be having the exchange of the genetic segments between the two homologous chromosomes, between the non-sister chromatids of homologous chromosome. This is really important to know, the non-sister chromatid. That means if you are talking about these two chromosome as the homologous pair, then the exchange will occur between the non-sister chromatids, not the sister chromatids. This 1 with 2 is sister chromatid with each other. This 3 with 4 is also sister chromatid to each other. So it will be always between the non-sister chromatids of homologous chromosomes, right? So this is a key event, and because of this particular event, the variation that we get, which is very important for evolution, comes over here. One more important thing over here to know is that by the end of this particular phase, what we are going to see is the completion of crossing over, right? This line is being depicted in NCERT, a very important line, that crossing over is going to be completed at the end of this particular phase, right? Now, because now the crossing over is complete, right? Exchange already ho chuka hai between the genetic segments. So now there is no more need of these chromosomes to be together, right? So now it is important that these two chromosomes segregate from each other so that they could be equally distributed into the two daughter cells. So that's when after pecitine we will be having diplotene, in which we are going to see the dissolution of synaptonemal complex. Right? So yes, Sagnik is correct over here. He is saying that diplotene में dissolution होता है synaptonemal complex का. क्यों? क्योंकि now the crossing over is complete. Now these two chromosomes has to segregate from each other so that they can be distributed in the two cells. Now after here, when the dissolution is being done, because recombination nodule is a very tight assembly, so even after this dissolution we will be seeing that the chromosomes are still attached, not across their entire length, but at certain points of crossovers, right? And those is the X-shaped chiasmata, correctly said by Ankul and Lucky. So those will be X-shaped structures, chiasmata, that will be still visible because these are the very compact structures. So entire synaptonemal complex will be dissolved, but those are the points where still the chromosome is attached. And even if this particular attachment is going to sustain, then again you are not able to segregate these two chromosomes into two daughter cells. That is the reason why now this structure also needs to be removed, and that is going to occur in the last substage, which is dikinesis, in which we are going to see the terminalization of chiasmata. Right? After the completion of this only, we'll be having, entirely separated homologous chromosome pairs. So this is what happens in the substages. Also, because dikinesis is the final substage of prophase 1, so other characteristic events like you have the disintegration, complete disintegration of nuclear membrane, then you will be having nucleolus disintegration, the disappearance of other organelles, aster formation, mitotic apparatus formation, all those things are going to be completed over here. That is the reason why we also call this final substage as the phase which shows the transition towards the next phase, which is metaphase 1, right? So this is what's going to happen, beginning with leptotene and ending at dikinesis. So with this information, if we get back, it is saying about the final stage, that means it is referring towards dikinesis, and we know that over here what is going to happen, correctly said, terminalization of chiasmata, which is depicted in option 3, is marked by the terminalization of chiasmata. The first one is recognized by dissolution of synaptonemal complex. This is going to be happening in the diplotin phase. That's why it's incorrect. Second one is saying can last for months or years in oocytes of some vertebrates. Again talking about diplotin. That's why it's one of the longest substages of prophase 1. Then fourth says involves nucleolus and nuclear membrane to reappear. Again incorrect, because this reappearance is a feature of telophase. We are going to see this both in telophase 1, in telophase 2, and also we are going to see, during the mitotic telophase as well. So this is again incorrect. So the correct answer for the particular question, the final substage marked by the terminalization of chiasmata. Right? I hope this particular question is clear to everyone. In case you'll be having any doubts, please post it on the chat box. All right. Meanwhile, let's go to the next question. And that's going to be question number 105. Talks about mitotic cell division is seen in the diploid somatic cells of, right? So mitotic cell division, right? Because this particular division is equational division, over there you are not going to see any, crossing over sort of a thing. So genetic content also going to remain the same. That is the reason why mitotic cell division is being adopted by organisms, so that they can have, cell repair, there's, growth thing. Even for unicellular organism, this is also facilitating reproduction part, right? So if we talk about these diploid somatic cells, these could be seen in both plants and animals, right? When we talk about only plants, only animals, that's not true. Both of them can accomplish this particular process for the process of cell repair, maintenance of body size, growth, right? For bacteria, because why it's incorrect for bacteria is because of the fact that when we talk about mitosis, right, this is talking about the M phase, so it is divided into karyokinesis and further cytokinesis, right? So karyokinesis means that over here the nucleus will divide first and then it is followed by the cytoplasmic division. When we talk about bacteria, we know for a fact that it is a prokaryotic structure, right? Prokaryotic organism where we don't see the well-defined nucleus as such. That is the reason why over here we are not going to see mitotic cell division. On the contrary, it will show amitosis, right? That is why this is incorrect. So yes, cell repair and to maintain certain cellular ratios, of course, mitotic cell division is important. So the correct answer for the particular question is going to be option number 4, right? All right. Now let's move ahead. another question, that's question number 108, which talks about we have to mark the wrongly matched pair for ETS of mitochondria. Okay? Alright, now let's talk about this particular one. So when we talk about the ETS of mitochondria, that means we are referring towards a pathway, referring to the pathway, which is going to talk about that how ATP formation is going to take place from the reduced coenzyme that's been produced in the entire process. Like in the glycolysis you produce it, link reaction, and then Krebs cycle, we produce a enormous amount of the reduced coenzymes, and now by using that we are going to produce ATP. So how that formation occurs is what ETS told us about. So this ETS also works on the principle of chemiosmotic hypothesis, right? So in chemiosmotic hypothesis, what all we need in chemiosmotic hypothesis, the first requirement to accomplish this is the presence of membrane, right? In ETS of mitochondria, that particular membrane is the inner mitochondrial membrane, right? This one. Now, the second requirement, the second and third requirements are going to be the presence of proton pumps and then further proton gradient, right? So in this particular case, if we see, then we can clearly see that there is a complex one, NADH dehydrogenase, which takes up the electrons during the oxidation of this NADH converting to NAD plus, right? And at the same time influx the protons in the intermembrane space, because of which in the intermembrane space the concentration of H positive ions is increasing, and as this H positive is taken up from the matrix, so over here the concentration is going to decrease. Now further same goes for complex 2. When we talk about complex 2, succinate dehydrogenase, this is the enzyme which also participates in the Krebs cycle as well. Only enzyme that is going to participate in both of them, and also the only enzyme of Krebs cycle that is embedded in the inner mitochondrial membrane. So over here it just takes up the electron but don't influx any protons towards the intermembrane space. So over here there will be no difference that's going to be created by this particular complex. Both these complexes, the first and the second one, gives it to ubiquinone, which transfer it to complex number 3, cytochrome BC1, which when takes up the electrons, right, also takes up the proton and influx into the intermembrane space, due to which again the concentration of H positive ions are going to increase over here, right? Further, the third one is going to give its electrons to cytochrome C so that it could be given to complex number 4, which again by taking the electrons from cytochrome C again influx back the protons in the inner intermembrane space. So here again the concentration of H positive increased, and because these protons are being taken up from the matrix, over there it is going to decrease, right? So finally what we have seen, multiple proton pumps, and because of which we have created a gradient. Gradient means that at a certain point the concentration is too high, then at a certain other point the concentration is too low. So by this we can clearly see that in the intermembrane space the concentration of proton has been risen up, while in the matrix region it has decreased, right? So this is how we have created the gradient. Now the fourth requirement that we have for the chemiosmotic hypothesis is the presence of ATP synthase, right? So after this particular one, the complex 4 is going to, because of this intermembrane space, this gradient has already been created. Now is the time to break it down and utilize its energy to create the ATP, right? So then again these protons are going to now be influxing back towards the matrix from this F0 particle, which is the transmembrane protein, and then this F1, which is a peripheral protein. Now when this influxing is happening through this, it will rotate the F1 particle, and this F1 particle will take up ADP and PI from one side and then release ATP. So this ATP formation, do remember, occurs on the matrix side, right? Not on the intermembrane space. Intermembrane space is being used so that we could create this proton gradient, right? So this is how the ATP is going to be produced with this particular ETS. ETS is a very important topic. Yes, you have correctly said there will be conformational change in both CF0 and CF1. We are not going to call it as CF0 or CF1. CF0 or CF1 is the denomination that we use for the chloroplast ATP synthase. Over here we are going to call it as F0 and F1, right, or FO or F1. So over here there will be conformational change in both F0 and F1. The conformational change in F1 is going to depict this particular formation of ATP from ADP and inorganic phosphate, right? So this topic is really important because multiple questions can come certain times in the form of match the column, where they'll be asking about the names of the complexes. Certain other times, you know, that what are the locations of each of them. Then certain times a very important one is these mobile carriers, specifically cytochrome C, where the location is, right? So this particular topic is really important with respect to the NEET examination, of course. So with this particular one, if we get back to the question, over here we have to mark the wrongly matched pair. So the first one is saying complex 2 is found in intermembrane space. So that's where it falls wrong, because complex 2, as we have seen over here, is present in the inner mitochondrial membrane. It is being embedded into it. So that is the reason why this is an incorrect option. So this is inner mitochondrial membrane, not the intermembrane space, right? The second one is saying complex 4 contains cytochrome A and A3. Right. So if we talk about complex 4, which is cytochrome C oxidase, a very important part about its constitution is that it will be containing cytochrome A and A3 and then 2 copper centers as well, right. So this is the major constitution of complex 4, because of which the electron tunneling is happening. So the question is saying that it contains cytochrome A and A3, so it's correct. Right, it contains cytochrome A and A3 along with 2 copper centers. Then third one is saying complex 5th is ATP synthase. This is also correct. Then option 4 says complex 1 receives electrons from NADH. That's also correct. That's why the name NADH dehydrogenase is there. Right. Complex ka sequence. Complex ka sequence is like when we talk about complex number 1 and complex number 2. They don't have any association with each other. They both give their electrons to UQ, which transfers it to complex number 3. Then further this gives it to cytochrome C, and this further provided to complex number 4. Now after this, these 4 are being required to create that, you know, proton gradient. After this, the complex 5th is being utilized to break that proton gradient. That's how the ATP is going to be produced, right? All right. So as we have to identify the wrongly matched pair over here, that's being depicted in option 1, right? I hope this particular question is clear to everyone. Okay. So in the meantime, let's talk about the next question, which says that the correct set of dominant traits with respect to flowers in pea plant which was studied by Mendel is, right. So when we talk about the Mendelian genetics, firstly he chose pea plant because of certain reasons. First, because of its availability, then the presence of the contrasting traits, then the presence of the short life cycle and have a huge number of progenies in one cross only, right. So when Mendel chooses character, he chooses 7 of them. The first one is the plant height. Then we can have the seed color and seed shape. Then we have pod shape and pod color. then we have flower color and flower position. These are the 7 of them, right? And if we talk about the dominant and recessive ones in this, right, then tall would be dominant with respect to dwarf. Then in seed color, yellow would be dominant over green. In seed shape, round would be dominant over wrinkled. Then in pod shape, inflated is dominant over constricted. In pod color, green is dominant over yellow. And in flower color, violet is dominant over white. And then finally in flower position, axial is dominant over terminal. Right? This is the basic convention. Now the important point over here is that as a generalization, what we do is we use the first letter, right, of this particular dominant trait as the name of the gene. Like if we want to use it for plant height, we'll be using capital T and small t gene. But this is not a headfast rule that you have to always follow it. It is general for the convenience and generalization that we use it. We could use some other name too, right? So these are the 7 characters that he has chosen for performing the crosses, and then performs like multiple amount of crosses like monohybrid, dihybrid, till heptahybrid. But with the purview of the NCERT, we have only 2 crosses, that's the monohybrid and dihybrid cross. So for those crosses, you should remember that what are the general phenotypic genotypic ratios, right? And also what kind of laws have been inferred from it. Like from the monohybrid cross, he derived law of dominance and segregation, while from the law of because of the dihybrid cross, he was able to comprehend the law of independent assortment, right? So these are the characters that he has took. So getting back from this, we have to choose the correct set of dominant traits. So over here, if we see violet color in axial position, both are dominant. So this is correct. Then talking about pink color in terminal position, pink color firstly is a recessive character, and pink color is not being observed in the pea plant. Usually if we see this sort of color, we see it in the case of snapdragon, right? Not here. Then we talk about white color in axial position. So white color is recessive, axial position is dominant, because both of them are not, so this is also incorrect. Then violet color in terminal position, over here violet color is dominant, but terminal position is recessive. That is the reason we can again rule out this particular option. So the correct set of dominant trait is depicted in option number 1, right? So this is how you'll be solving this question. This is a very easy question, but comparatively you will be finding a bit of difficult questions in terms of crosses. So you have to practice those type of crosses, that how you are going to analyze the results. Okay. So now let's move ahead to another question, which is question number 113, and it says which one of the following genotypes can produce maximum 8 different type of gametes, right? So when we talk about the types of gamete, so types of gametes can be calculated with the formula 2 to the power n, right? So over here n is going to be number of heterozygous loci that's been present, right? So if we see the very first genotype over here, that is capital A small a, capital B small b, and small c, right? This is what's been given. So over here if we see, this is a heterozygous loci and this one is. This is homozygous, so we are not going to consider this one. So over here, because the number of n is 2, so this is going to be creating 4 of them, right? So here we are going to have 4 of them. Now in the next genotype, if we will see, we'll be having this as the heterozygous loci A, B and D. C is not going to be considered. So over here if we calculate, then it will be 2 to the power 3 makes it 8. Then we'll have to see here we'll be having capital A small a, capital B small b, capital C small c and capital D small d. All of them are going to be heterozygous, so it will be 2 to the power 4. So this comes out to be 16. Then if we'll see this one, capital A, capital B, all of them are homozygous, so we are not going to consider either of them. So it makes 2 to the power 0, which is equivalent to 1, right? Now the question over here is saying that which one of the following genotype can produce maximum 8? It can produce 8, but it's much beyond than that. Maximum 8 would be produced by this one. So the correct answer over here is going to be option number 2, right? If it will be like minimum 8 number, then it could be this and this both. But here the question explicitly say about that maximum 8 number of, different type of gametes could be produced. So that's why the correct answer is going to be option number 2, right? It's not 3, it's option number 2, because of the maximum 8 different types of gametes are going to be produced by this genotype only. It is beyond 8. That's why it's an incorrect option, right? Okay. Is it correct? Is it like being understood by everyone? All right. So now let's go ahead with another question which talks about that the pure line pea plant for green pod color was crossed with pure line yellow pod colored pea plant. The pod color in F1 generation will be, and it can be explained by, right? Okay. So when we say that the pure line pea plant for green pod color, right, as we have already discussed, this green pod color is basically the dominant trait, right, and it was crossed with pure line yellow pod color. This yellow pod color is recessive trait, right? So if this particular cross is being done, so let's suppose the parents over here, we are going by the normal generalization. So because green is dominant in pod color, so that is why we are taking G as the gene symbol. So this is having green pod color. Then it is being crossed with pure line, right? This has been explicitly mentioned about pure line. Pure lines are the ones that are homozygous for a particular trait, right? So not for all the traits, but for the particular trait that's been in the question. So it's been pure line for the pod color, it's green, and this is yellow, right? Now if we are making gametes, a single type of gamete will be produced, that will be capital G, and over here a single type of gamete will be produced, that will be small g. Why? Because when we talk about 2 to the power n, this n means the number of heterozygous loci. Over here both of them are homozygous, so this will make, it will be 2 to the power 0. So one type of gamete will be produced, that will be capital G for this plant and small g for this plant. Okay. So now when it will be fertilized, it will be capital G and small g, which is again going to yield the green pod color, the F1 generation, right? So this is what's been asked, the pure line pea plant for the green pod color, and it was crossed with pure line yellow pod color pea plant. Now the pod color in F1 generation, so the F1 generation that we have seen here is having green pod color, right? So with this we could simply eliminate these two options, because they are saying that in the F1 generation the recessive phenotype is going to be observed. So the ones that we are left with is second and third, right? Now this could be explained by, so this could be explained by law of dominance, not because of law of segregation. Why? Because when we talk about law of dominance, which was derived from monohybrid cross, it ascertain the fact that firstly allele, by the time of Mendel it was not allele because he was not sure of the physical existence of that particular thing. So he named it as a factor. So firstly the factor occurs in pair, right, and then further there will be like for a particular factor there will be two forms. One is the dominant one and one is the recessive one. Dominant one is the one that is going to express itself whether present in the homozygous condition or heterozygous condition, but recessive is only going to express itself in the absence of the dominant one, that means in the homozygous form only, right. So over here also what we have seen that in this particular case, because the dominant alleles are present, that's why the phenotype that they have exhibited is the dominant one, the green pot color. Even if it is present in the heterozygous form, it is being producing the same dominant phenotype. On the contrary, when this recessive allele is present in the homozygous condition, that means there is no companion dominant allele over here, only then it will express itself in the yellow pod color. ठीक है. अगर दो पेरेंट्स डिफरेंट जगह पर तीन हेट्रो, तो zygote कैसे निकालेंगे? Just to multiply the number of gametes with each other, right? That's how you will be getting the number of zygotes. Like in this particular case, 1 into 1, it will be simply 1, right? That's how. So if you will be having heterozygous loci, you can either make the gametes and then multiply them, or you can do it with this particular formula as well, 4 to the power n, right? So this is how we could explain it with the help of law of dominance. Law of segregation is also being derived from the same thing, that's monohybrid cross, but law of segregation talks about that during the time of gamete formation, we will be having segregation of the allelic pair, right? There will be no blending of the character. So segregation is going to occur during the gamete formation. So this particular one is correctly depicted in option number 3. So that's given over here, option 3, all else could be ruled out, right? Ankul, is your question is clear to you or not? All right, so you can put in the chat box, okay? So over here, this particular question is, the correct answer of this particular one is given in option number 3. This is how you will be solving this question. Moving ahead, let's talk about another question which talks about assertion, says that dominance is an autonomous feature of a gene, right? So this is where it is incorrect. Dominance is not an autonomous feature of gene, because when we talk about dominance, as we have seen over here, so in that particular case, it is also going to be depicted type that how exactly, okay. So I am saying dominance is an autonomous feature of a gene is incorrect because of the fact that the dominance is not an autonomous feature. Why it is not an autonomous feature? Because when we talk about dominance, it's not only going to be depending upon a particular trait, right? For one trait, this particular allele could be dominant, and for other it could be recessive, right? In the pleiotropic genes, we could see it. Then depending upon the conditions also, it will be changing. Like certain times when the body needs it, the gene has to express itself. So it is, having the dominant form. Then in some other case, it's not at all, suppressing, even if it's the dominant copy, right? Like you could see in case of the black operon, right? That till the time lactose is not going to be present, then ZYA cannot be able to express itself, right? You could see in other cases also, in the case of synthesizing, amino acid synthesizing genes also. So dominance is not going to be depicted by the gene itself, but by the conditions also, but by the trait under consideration also, but by the availability of the resources also. So all these things are going to decide the dominance of a gene. So that is why it's not an autonomous feature of a gene, right? So this is the reason why assertion is going to be incorrect, right? Then we have reason. Dominance depends much on the gene product and the production of a particular phenotype from this product. So yes, it does depend on the gene product and also the particular phenotype from this particular product. So this particular statement is correct, and because of this, we could say that assertion is false but reason is true. Both are not false, so we could rule out this one and this one. Okay, so Ankul, as you have said that you have given these two particular genotypes, you can find the number of gametes, right, and then multiply them. Simple as that. This is what you have to do. Find the number of gametes, whatever number of gametes, like you have said, the genotype that you have given. In the first parent, it is being heterozygous at 3 loci, then multiplied with, you have the second one is being at the 4 loci, right? So 2 to the power 3, 8, 2 to the power 4, 16, multiply it, right? 128 would be the answer. Right? Is it clear, Ankul? Number of gametes you have to identify and then just simply multiply it, because the fusion of that is going to decide that how many types of different zygotes you will be having. Okay? Alright, so getting back to this particular question, assertion over here is a correct statement which talks about that it is an autonomous feature. On the contrary, the second line is correct, that it is dependent on various factors, right? So this is how you'll be having assertion and reason solved, right? Now moving ahead, let's have another question which talks about the biofertilizers, right? So when we talk about these biofertilizers, that means we are talking about the organisms, the biological organisms itself, which are going to increase the fertility of the soil by adding certain nutrients to it, right? Like for example, if we take the example of azotobacter or azospirillum, right? Both of them can add up the nitrogen, because these are the nitrogen fixing bacterium, right? So both of them can add that particular one. Then we could see the case of glomus, which is a fungal genus which forms the mycorrhizal association and then can absorb, you know, phosphorus for the plants, right? Then Anabaena, which is a cyanobacteria, would be a very good biofertilizer for the case of paddy fields, right? It's a very good biofertilizer in that particular case. We could see certain, you know, symbiotic associations with bacteria like Rhizobium. This is a bacteria that can form a symbiotic association which can, you know, add up the nitrogen for the plants, can fix that particular one. So these are the biofertilizers. Now when we talk about Bacillus thuringiensis, this is not a biofertilizer. On the contrary, it is a biocontrol agent. That means when we are going to use the organism, right, when we are going to use this particular organism, we can control the butterfly caterpillars, right? So when we talk about the genes are used, see, this particular one can be used in its natural form because it is being, you know, comes in the form of dried sachet also, right? In biotechnology, you can use the genes to make the plants resistant for it entirely, but you could use it as a spray also, as a normal biocontrol agent in organic farming, right? Where you are going to use entirely the biological resources so that you could control them, right? Control the pests and the pathogens to a manageable level. Because if you are going to, you know, what you could say, delete them from the environment, then what will happen? The ecological balance is going to be disturbed, and one way or the other we have to pay for the consequences. So that is the reason why we use this particular bacterium in the form of dried sachet for the control of the butterfly caterpillars, right? Correct. For brassicas we could use it. So and that's why this particular one is also used in you know, recombinant DNA technology also, because this particular one has the capability to you know, control certain pathogens. So that is the reason why certain insects, not pathogen, pests. So that is why it is used more like both in the natural form also and in the genetic engineering in the form of donor also, right? So this is a biocontrol agent because we have to find the odd one out with respect to biofertilizers. That's why the correct answer is going to be option number 2. Rest all could be used as biofertilizers only, right? Moving ahead. We have the question which talks about which of the following are the most important climatic factors that regulate the rate of decomposition. In the chapter ecosystem, decomposition is a very prominent one. From ecosystem, we get the questions from energy flow, and from this particular topic also, decomposition. So when we talk about decomposition, the ulterior motive is going to be we have to break down the bigger complex particles into the smaller ones, right? For that, initially the detritus that's being available is going to be fragmented by the detritivores, further from that, certain component is going to get wasted because it's going to leached out into the water table, right, from the soil, and then the other part is going to be acted upon by the decomposers, the fungi and the bacteria. They'll do the catabolism of that, and from that again two type of sections that we are going to receive. One that's easily digestible, to which mineralization is going to be happen easily, and then there will be other one which is really difficult to digest, forms the humus. Humus again gets digested, but it will take a huge amount of time to digest it, right? So for this particular process, decomposition, a very important thing is that it's an oxygen requiring process, right? Now as over here they are asking about the climatic factors, right? This is really important. So when we talk about the climatic factors, the first factor is going to be temperature, you know. So usually also we know that whenever there will be high temperature, the rate of reaction is going to be faster. So over here also decomposition is going to be speedy if the temperature is going to be too high. If it is low temperature, it's gonna lower down the process of decomposition. Then talking about soil moisture, this is again a very important climatic factor. Now for the soil moisture, it should not be low, it should not be high also. In both the cases, it's going to impede the process of decomposition. It should be at the optimum amount in order to fasten the process of decomposition. Over here it says soil moisture and soil pH. We could rule out this particular one. Why? Because soil pH at certain times it has a role, but not that much implicit role as the temperature is holding, right? Then aeration and temperature again we could rule out, because when we talk about soil pH and aeration, both are edaphic factors, not climatic factors, right? Then we have soil pH and aeration, edaphic factors, not the climatic one. Here one climatic factor has been included. So क्योंकि question में हमसे climatic factor के बारे में पूछा है, so the most important one is going to be soil moisture and temperature, right? So for decomposition, you should also know that in which conditions it is going to speed up and in which condition it's going to be impeded out. Like if we'll be having high temperature, if we'll be having proper aerobic conditions, if it will be optimum soil moisture, if it will be easily digestible detritus, like if it is rich in, you know, nitrogen sugars or water soluble, you know, substances, then the decomposition is going to fasten up. If on the contrary, if we see anaerobiosis, that means absence of oxygen or lack of oxygen, or we'll be having low temperature, or we will be having detritus that's going to be rich in lignin, tannin, substance like these, or we are having low or very, very high moisture, right? In these conditions, we are going to see that the process of decomposition is impeded up, right? So this is how you should also know about that, where it's gonna be fastened up and where it's gonna be reduced, right? So this is all about this particular question, and for this particular question, the correct answer is going to be option number 1, right? Now let's move ahead to another question, which says that how many mitotic divisions will take place in the formation of 32 daughter cells from one cell, right? So for this particular one, the formula that we'll be using is n minus 1. N over here is the number of daughter cells that we receive. So if the number of daughter cells will be 32, so the number of mitotic divisions will become 31, right? So that's correctly depicted in option 3, 31. Rest all would be ruled out simply like this, right? So the correct answer is going to be option number 3. This is a really easy question, very simple formula, n minus 1, whatever daughter cells, number of daughter cells you have, just subtract 1 from it and you will be receiving your answer, right? Okay, so multiple students are giving correct answer, very good. All right, now let's move ahead. We have another question, which is that in mitochondrial ETC, which carrier molecule takes electron from both complex 1 and complex 2 and delivers them to complex 3, right? We have discussed the ETC. So we know that when it will start with complex number 1, right, which is NADH dehydrogenase, which takes up the electron from NADH. And then we have complex 2, which is succinate dehydrogenase. Right? Both of them give its electron to ubiquinone. Now, why this particular ETC is so important? Because of the fact that because of these two only, the oxidized coenzymes are being produced again, right? And those oxidized coenzymes are important for the continuation of the Krebs cycle, right? And also for the continuation of link reaction and glycolysis as well. So these are basically the resources with which it is going to be supplied back to those particular cycles, right? So C1, that's complex 1, is going to give it to ubiquinone, complex 2 also going to ubiquinone, and these two doesn't have any interlinkage in between, right? This is going to influx the protons in the intermembrane space. This is not going to do that. Then from ubiquinone, it is going to be given to complex 3, which is cytochrome BC1 complex, right? This will give electron to cytochrome C. The location of cytochrome C is also very important. When we talk about the location of cytochrome C, this is being present on the outer side of inner mitochondrial membrane, while ubiquinone is, traversing across the membrane only. So cytochrome C is the mobile carrier that's going to give it to complex number 4, which is cytochrome C oxidase. Right? So this is how the complexes are working in association with each other so that they can create a gradient of protons, and finally when it has been broken down, we could generate the ATPs from it, right? So with this, if we get back to the question, it says that we have to talk about the carrier molecule that takes electrons from both complex 1 and complex 2 and delivers it to complex 3. As you could see clearly over here, that's ubiquinone, right? So this has been given in option 4, so this is correct. When we talk about first one, cytochrome C, this is giving the electrons from complex 3 to complex 4, right? Then ferredoxin, again incorrect. Ferredoxin is we used during the non-cyclic photophosphorylation, right, which transfers the electron from PS1 electron acceptor to FNR. Then plus 2 quinone again is a carrier which is going to be there used in the non-cyclic photophosphorylation again. So these are not being involved here. So that is why the correct answer for this particular question is going to be option number 4, right? Correct. So it's been used during light reaction. Now moving ahead to another question which talks about we have to select the mismatched pair. So the first one is saying complete dominance in pod color in Pisum sativum, right? So Pisum sativum, that means we are talking about the garden pea. And when we talk about the pod color here, over here we'll be seeing two traits. One, that's going to be green. This green is dominant over the yellow, right? So this is recessive one. So if we talk about the pod color, if this particular allele, that's capital G, is present, whether in homozygous or heterozygous condition, it will express itself, right? So this is an example of complete dominance where a gene completely imparts its dominance over the other. The second one is saying codominance, ABO blood group in humans. This is again true. So when we talk about blood group in humans, it will show complete dominance if it is having genotype IAIA or IAI, or having IB, IB, IBI, or having this particular genotype, small I. But if it is IAIB, it is showing codominance. That means both the alleles are equally dominant to each other. So this is also correct. Then third one is saying multiple alleles exemplified by phenylketonuria. This is incorrect. Phenylketonuria also has this complete dominance thing, but on the contrary, it is a pleiotropic gene which manifests and control multiple phenotypic expressions, right? So this is incorrect. Multiple alleles, if you want to see the example, you could take the example of ABO blood group which have 3 alleles, in contrast to the Mendelian proposition which says there are only 2 alleles for each of the character. Then pleiotropy says gene controlling starch grain size and seed shape in P. So this is correct. The gene that controls the starch synthesis in P is going to control both the starch grain size and seed shape, and the pleiotropic gene is the one that one gene that controls multiple effects, right? So that is why this is a pleiotropic gene. Okay? So this is how we will be having the we have to identify the mismatched pair. So the mismatched pair over here is exemplified in option number 3. So that's the correct answer, right? So this is how you will be solving the questions related with these chapter. So I hope this particular session is useful to you. So please keep practicing and keep focusing on NCERT. Thank you very much.